Hello everybody, my name is Joseph with discussion.science.blog. How are you doing today? Well, that's fantastic. So today I'll be talking to you about Anammox bacteria. Now, for those of you that don't know what Anammox is, it is anaerobic ammonium oxidation. So in other words, it just gets ammonium and nitrite and produces nitrogen gas and a little bit of water. And Anammox bacteria just does that process. So let's have a look at the history of Anammox bacteria, shall we? So picture this. It's a beautiful lake, Lake Mendota in Wisconsin, and it's 1932. So there's unusual amounts of nitrogen gas being produced here, and now this water is hypoxic. So there are denitrifying bacteria in here producing some nitrogen gas, but there's a lot of nitrogen gas being produced. So there's some other thing that's producing nitrogen gas, and we're not really sure what that is. Big question mark, am I right? Yeah, so flash forward to the 60s, 1965, a man named Fred Richards noticed a lot of the ammonium which should be produced was missing in his study. So in other words, the ammonium which should be there was being converted to something else. Now he didn't know what that was or what was happening, but it was actually the Anammox bacteria. That is the study. Now the specifics are unimportant for this presentation. So flash forward to 1977. A man named Angelbert Broder predicted two chemolithic autotrophs capable of converting ammonia to nitrogen gas through thermodynamic calculations. Now, the specifics of those calculations are unimportant, but those interested, that is a study which he predicted those two chemolithic autotrophs. Alrighty, so now let's flash forward to 1995. A man named A. Mulder completed this study, and within that study, his observations were consistent with those of Engelbert Broder's calculations. So there was an anaerobic denitrifying bioreactor. So denitrification, it doesn't involve ammonium. Now the influent is water going into the wastewater treatment plant, and the effluent is the water coming out of the wastewater treatment plant. Now as you can see, at about day 90, the ammonium concentration on the effluent starts decreasing and almost gets depleted. Essentially what is happening within this wastewater treatment plant is the ammonium is being converted to something else, but it was a denitrifying anaerobic bioreactor. So ammonium concentration shouldn't have been affected, but it was. So that is the first identification of the Anammox bacteria. And now let's have a look at a correlative study involving Anammox bacteria. So there was a section within this study that aimed to figure out how the Anammox bacteria did its thing, how the metabolism worked. So what they did is they got an anoxic bioreactor and they left it for an entire year because the Anammox bacteria take quite a while to reproduce. Now within this anoxic bioreactor is synthetic wastewater ingredients, so bicarbonate, ammonium and nitrite, and these are very common ingredients in the wastewater influent. Okay, and now they used fish to determine it was dominated by a species called K. stuttgartiensis. So they obtained the genome of this bacteria by using shotgun sequencing as the scaffolding for the genome and then filled in the gaps using bacterial artificial chromosome libraries. And what they found is they found 200 genes relating to Anammox catabolism, and they did this by focusing on domains of interest. So they looked at the sections of the genome that had to do with electron donation and acceptance, which is a common characteristic of the Anammox process, and specifically these sections were the heme ion binding sites, which also donate electrons and accept electrons, etc. And they also found a functional homologue to nitrite reductase, which converts nitrite to nitric oxide. Now this is confusing because nitric oxide wasn't considered an intermediate at the time of the study. So as you can see, this is the diagram from the study, and the little red box, the NIR, represents the NIRS enzyme stated at the bottom, which is the functional homologue. And as you can see, it converts nitrite to nitric oxide, the intermediate, and the new Anammox pathway that they discovered. It then goes on to be converted to ammonium, then to hydrazine, which is another intermediate, and then finally to nitrogen gas. So, in summary, for this study, they discovered something that was not compatible with the previously postulated model for Anammox catabolism. It was a new pathway, the nitric oxide pathway. It also conforms to the Occam's razor, which is pretty much the path of least resistance in the biochemical world. Now, this was correlative, so they didn't actually experimentally validate anything. They just got their genome and compared it to string databases online. But we also know that correlation studies can be good because it leads to more studies which are causative, and the results of that study are currently being experimentally validated. Alrighty, so now let's look at a causation study in terms of Anammox bacteria. So do Anammox bacteria rely on other biochemical processes for ammonium supply? In other words, it's asking, where does the Anammox bacteria get its ammonium from? Does it produce its own ammonium? Or if there's already ammonium present, does it use that ammonium? This 2007 study aimed to figure that out. So what they did is they got anoxic excitaners and they filled it with C-formate, which was the electron donor in the Anammox process, isotopic N15 nitrogen in the nitrate. So it was essentially the nitrogen in the nitrate was labeled. Okay, that's what we need to know, really. And then alongside that, they had a non-labeled compound, either nitrite, either ammonium, or either nitrous oxide. Now, the non-labeled compounds acted to pull the N15 isotopes when they were converted. So if the N15 from the nitrate was converted to the nitrite, then those N15 nitrite compounds would then be pooled with all the rest of the non-labeled nitrite compounds. Okay, now this is their results from that study. The y-axis is the percentage of the original N15 nitrogen from nitrate. And as you can see with this red box, this shows that about 40% of that original isotopic N15 was converted to nitrite. So it's saying that some of the nitrate has been converted to nitrite. And that is only possible through the Anammox bacteria because there's nothing else at play. Now these two bars just essentially tell us that no matter the concentration of ammonium, the Anammox bacteria is still producing its own ammonium. So as you can see down the bottom right, the cross-hatched section of the bar, that shows that there is N15 ammonium, which means the nitrogen was converted to nitrite from the nitrate, and then eventually to the ammonium, regardless of the abundance of the ammonium already present. Okay, now this last bar is just to make sure that it isn't in fact a denitrifier. So there's no sign of nitrous oxide, which is a compound in the denitrifying process, and so there's no N15 nitrous oxide, which is great, really. Okay, so essentially in this study, we were able to capture each step of the process using pools of the non-labeled compounds. We were able to determine that K. stutgartiensis is Anammox, and it does produce its own ammonium, no matter the concentration of ammonium already present. Now, this study was indeed causative. Booyah, baby. That is a causative study. That is great. That is fantastic. Okay, so in summary, we just looked at the Anammox bacteria history. We also looked at a study of Anammox bacteria, which is correlative. And then we compared it to a study of Anammox bacteria, which was causative. Thank you for listening. Have a great day.